wonderful to have us back on the channel, and it's always wonderful to have us together discussing science. Today, we are going to discuss a unique concept, and we're discussing the concept of plasmids. What are plasmids? Now, plasmids are what we refer to as extra chromosomal DNA. They are circular DNA that are present in a lot of cells, both fungi, archaea, bacterial cells, even some eukaryotic cells. We have plasmids present in these cells. And what do plasmids do? Plasmids, they are unique because they exist apart from the normal chromosomal DNA. That's, they exist away from the normal chromosomal DNA. And they are there, they confer a lot of characteristics on these cells, and they do a lot of things that are unique. So we'll be discussing plasmids in details, what they do, what they are, their different types, and the way they affect the cells in which you can find them. Now, when you talk about plasmids, there are different types of plasmids. And like I said earlier, plasmids are they are DNA that are extra chromosomal. They are not part of the main DNA of the cell, but their presence there, confers particular characteristics on those cells. Now, let's talk about the different types of plasmids. There are many, many different types. There are about 4 different types of plasmids. The first one, we refer to them as sex plasmids, which sometimes we call them fertility plasmids or F plasmids. Some people even call them conjugative plasmids. Now, these plasmids, they are unique because primary function they do is that they help bacteria or any other kind of cell to be able to go into conjugation. So we know that normally bacteria divides by the process of binary fission. But the presence of plasmids in bacterial cells helps the bacteria that they're able to undergo conjugation, meaning that 2 bacterial cells can come together, create what we call a sex pilus or pilus, which is a passage between the 2 bacteria. And then when they create this passage, there's the exchange of genetic materials from one bacteria to the other, vice versa. And the sex pilus or the conjugative pilus or the F pilus or fertility pilus, any name you want to give it, is the one that facilitates this process. So it's a very important type of plasmid. Now, also we have the R plasmid, which takes its name from resistance. So the R plasmid is present in some bacteria, and the R plasmid carries the genes, carries some genes that code for antimicrobial resistance. So thereby conferring, resistance on the bacteria. So in simple layman's term, any bacteria that is carrying an R plasmid has a plasmid that is having some, circular DNA that has genes that code for antimicrobial resistance. And so, by virtue of the fact that that bacteria has that plasmid, that bacteria is able to, have resistance, be resistant to particular bacteria. So, there's fertility plasmid, there's R plasmid, there's also call plasmid. Now, there are some plasmids that help bacteria, to be able to code for certain proteins that produce or are components of toxins. So we refer to them as call plasmids and call plasmids, sorry. And they are able to, help to, for example, there's what we call bacteriocins. So these are, proteins that are produced in bacteria that have toxic property, can be looked at as toxins. So the presence of these call plasmids are there to help, and they push out this kind of proteins from within bacteria. It's also important to note that some bacteria have what we call virulence plasmids. And virulence plasmids, just as the name implies, they carry virulence genes that code for virulence factors, thereby conferring or making sure that the bacteria has a virulent nature because it has the presence of these plasmids. So these plasmids are important. Majorly 4 plasmids which are important for us to know: fertility plasmid, call plasmid, resistance plasmid or R plasmid, and then our virulence plasmid. Then we'll wrap up this class by discussing some of what are some of the functions of the plasmid within the bacterial cell. What are some of the functions of the plasmid within the bacterial cell? It's important to know that the plasmid can confer on the bacterial cell resistance. So it can make a bacterial cell to be resistant. And resistant to what? Resistant to antibiotics or different types of antimicrobials. So this is one of the functions of plasmids. Plasmids also help bacteria to be, or even remember I told you that the plasmids can be even in eukaryotic cells. So they help any cell that they are in, they help them to be able to metabolize different kinds of substances that normally the cell will be unable to metabolize. Substances such as heavy metals, such as hydrocarbons, you see bacteria being able to metabolize those substances, meaning that they're able to work with those substances, they're able to digest those substances within their structure. And this is possible due to the presence of plasmids. Another important role that plasmid plays is that it helps a bacteria or any other cell to become really pathogenic in nature. And this is the presence of our core plasmid, the presence of our resistant plasmid, the presence of our virulence plasmid will make a bacteria to be really, really pathogenic in nature. Lastly, in biotechnology, it's important to understand that the plasmids help, they act as a vector, a viral vector, sometimes a vector for different processes that are being run in genetic engineering. Plasmids can act as a vector, and so they can be used, to transfer genetic materials from one bacteria to another, or to even put new genetic materials into a bacteria for the first time. Thank you for listening to this our short lecture on plasmids. We have a second video on plasmids too. Check that video out where we look at the mechanisms of plasmid transfer. So check that video out, and I hope this helps you out in your school and in your science. Have a beautiful and wonderful day.